So we're going to go into the excitation-contraction coupling, but we're really going to add, here's the nerves. The nerves, how do we get the ball rolling? Here's a brain, here's a nerve coming off, going to a muscle, and then like it would, move. Skeletal muscle, remember, it's voluntary. So you have to have a thought, like, I want to move my arm. There you go. So you have to think about it. Where other types of muscles, we don't. This is a single neuron. A single neuron could have little ends so that it will contract several muscle cells. A group is called a motor unit. So from a single neuron. So this would be a motor unit of like 1, 2, 3, 4, 8, 7 muscle cells. But if you're in the quadriceps, we don't really need fine, light motion. So a motor unit could be 100 muscle cells. And if I just want to tap something really lightly, and then maybe I just want to kick something really hard, I might have several neurons with several additional motor units that my brain would would recruit. An example too is like our life experiences sort of tell our brain what to expect so we can anticipate how much muscle we might need. For instance, say like a jug of milk, like a carton of milk in your fridge. I remember as a teenager, when my mom wasn't looking, and I would go get the carton of milk, and uncap it, drink it. Well, back then they had the foldy out thing, which made it even grosser, actually, Publix. But, you go grab the milk, and I would just drink it from the fridge when my mom wasn't looking. But I have a brother who liked to do that too, apparently. So I remember I'm going to the fridge, open it up, and I grab it, and kind of what a carton of milk would weigh, right? And I knew my mom had been in the grocery store, so it's going to be a new one. So I grab it, but my brother beat me to it. He drank most of it. And so I grab it, pre-recruiting many motor units, and I just launch the sucker across the kitchen. Because so part of it is the voluntary nature of our pre-recruitment. It's about what's coming from our brain to our muscles. Yeah, so I had a mess to clean up. So anyway, but it's part of life. So that's recruiting. So your brain can choose, so you only need a couple, maybe a small motor unit versus a large. So again, this is just going back to the voluntary nature and really what a motor unit. So this is an example of what we're going to get to, spinal cord in this unit. So that's coming up as well. But say we have two different single neurons coming off the spinal cord, and this will make sense later, but coming out, going out to the muscle. But you can see, neuron blue has these guys, and neuron red is recruiting these. So it's just within a muscle, but each muscle does have its own little activation point from a neuron. But it doesn't have to be one neuron to one muscle. Usually like a single neuron, and it's going to do several muscle cells. That would be called a motor unit. There are large motor units like in the quadriceps, but then in your forearms and your eyes, one motor unit could be just a couple muscle cells because you have really fine motion that you want to have more control. Here we are. We're going to go into, say, whatever neuron we got dealing with. Let's deal with one end part to one cell. A single neuron, so that's what this is, the end of a neuron. They got these weird things around it too. They look like hot dog buns, but we'll deal with the hot dog buns later. So here's the neuron coming out, and the critical element is it goes to the muscle. It doesn't touch it. It like hovers. There's a space. This is an important component coming up. So here's the neuron, and here's the muscle cell that we're already familiar with. So this is known as the neuromuscular junction. So it's kind of a big word, but when you break it down, it's pretty easy what it is. Nerve going to the muscle. They don't touch, they hover. And then this is how the nerve is going to ultimately tell the muscle, it's go time, time to contract. So here's the electrical stimulus. A nerve sends an electrical stimulus to a muscle. At the end of the nerve, so some neuron anatomy I'm giving you ahead of time, that's known as the synaptic terminal, that's an axon terminal, synaptic bouton. There's actually lots of names for this little end, bulbous end of the neuron to the muscle. So as we look into this section, we're going to zoom in on the neuromuscular junction. Okay, so can you see that we're zoomed in? The yellow part is still the end of the nerve, that bulbous end. The red area, that's the sarcolemma. See how it dips down? It kind of makes a little cup area where the nerve goes, but see how they don't touch. There's still the gap. The synaptic cleft, I was just kind of scribbling the space. That's a space between the two. So the synaptic cleft is the gap between the end of the neuron, and it's between the end of the neuron and the sarcolemma. The vesicles are these little pouches of green dots. Vesicles contain neurotransmitters. Because we're talking about muscle, it's the same neurotransmitter across the whole body, acetylcholine. We're going to learn next week that there's other neurotransmitters. Many of you guys know about dopamine, norepinephrine, serotonin. If someone's on Prozac, it's a drug that targets serotonin. And so anyways, neurotransmitters could be lots of different chemicals. So in a general sense, you have the end of a nerve sending out these little chemical beads to activate the next thing. For muscle, it just always will be acetylcholine. That's the particular neurotransmitter here. But we have these little pouches called vesicles. When it's go time, it's going to spring it to the end, and they dump out these little acetylcholines, and it's going to go to the sarcolemma, and then now the muscle knows it's time to get busy. Here comes the action potential. That's the electrical signal coming down the nerve. That's known as the action potential. That's going to be, we'll go into that detail next week. It then releases those little green dots, acetylcholine, the neurotransmitter, into the synaptic cleft, goes across, and then now it's going to go to the other side, and it actually causes, if you're writing this in notes, I don't care that you know this term now, but you must know the term next week. So for your complete notes, you can write, it causes depolarization. When you see the pluses, when a cell becomes more positive, it's depolarizing. It's depolarizing or activating it right under where the acetylcholine went, where the neurotransmitter went. But it's going to then radiate out. Neighbor area is going to then depolarize its neighbor area, which is going to depolarize its neighbor area. So that will continue on over to the sides and down the T-tubules. So this is how a neuron from the outside of a muscle can send acetylcholine, depolarize or activate the membrane right under the nerve. But then once it does it, it's like it infects it. Its neighbor depolarizes its neighbor spot, which depolarizes it. And the whole point of the T-tubules is it brings that activation process into the inside of the cell. So instead of it just running down, I should have made it like where it was lit up one, a plus, and then a plus, and then a plus, and then a plus, because it's really like dominoes. One area hits, causes the next area to become depolarized, causes the next area. So it does it more sequentially than just the sliding thing I just showed you. Where the T-tubule comes down to the sarcoplasmic reticulum, this area is known as a triad. It just means where the T-tubule comes in. A neuron sends a neurotransmitter, depolarizes or activates the muscle cell, that depolarizes or activates the T-tubules, goes down into the center, all throughout a muscle cell, and the T-tubule tells the sarcoplasmic reticulum, let the calcium out. This slide just lets you know sarcoplasmic reticulum just has the calcium. So we already know that. So it's just the calcium pouch. We're uncovered the sarcoplasmic reticulum, and we're looking underneath the sarcoplasmic reticulum, and now what do we see? The myofibrils with the myofilaments. And we're back to the actin and myosin. Nerve sends the stimulus down, releases acetylcholine or the neurotransmitter, crosses the synaptic cleft, just activates, depolarizes the sarcolemma or the muscle cell. That gets transmitted down the T-tubules so the whole muscle cell can get activated all at the same time. It goes to sarcoplasmic reticulum and releases calcium. Calcium and contraction. So you guys already know this, but we're just putting it together now. So here we are, our actin and myosin. So we've got myosin and our actin. And now the next thing is just showing you one of these little sections. So we've got where it was sarcoplasmic reticulum, had calcium. We're going to peel that away so we can look under it is now, you know, the actin myosin. So then this next one is sort of zooming in on a small section. That's your sarcoplasmic reticulum above. This is our actin. Remember, it's got the tropomyosin and the troponin. And then this is part of the myosin, the furry thing. So we're just going to go through the pink dots now are going to be calcium. This is at rest. Calcium's inside the sarcoplasmic reticulum. Tropomyosin is covering up the active sites on actin. Troponin, it's got nothing bound to it. Cross bridges of myosin are just laying down. They're set. It's like they're ready to go, but they don't have anywhere to go because the active sites on actin are covered up, right? So this is a relaxed muscle. So as soon as calcium's released, this is the whole, from this point, calcium inside to calcium being out, the T-tubule. That's what did it. So remember, nerve, the activate muscle goes down the T-tubules. The T-tubules say, sarcoplasmic reticulum, open the gates. And the gates are open. And now what leaves? Calcium. You know, I grew up on a ranch, so I think in terms of horses and cows. You're either in the corral or you're out of the corral. You know, so you have your gates. So in this case, the gates are open, the calcium gets let loose. In the cell, calcium will only want to bind to what? So here we are, calcium is now free, let loose, kind of around the myofibrils. That's why the blue things were around them when we did the cell anatomy. So now you can see the troponin, now it's pink. And then what happens as soon as calcium binds to troponin?
It moves.
There we go, tropomyosin moves out of the way. Our yellow active sites are there. Now what's gonna happen? Myosin lifts up, it's happy because it sees its active site. As soon as it binds to its active site, what does it do? Cranks over. Remember the crank? And then watch actin shift over. Lots of them are doing them at different times. So when one of them is coming to reset, it's not like you let go and then the actin goes back the other way because another one's hanging on. So they're all like, it's like a tug of war. You got people, and you know, when you're ungripping your hand to do another, someone else has the grip on, you know, so that's really what's happening here. But we don't really show it because we show them all releasing at the same time. So now we have the contraction. The myosin cross bridge will stay bound to actin until we detach. How do we detach? ATP comes in. Now ATP is bound. It's going to then, what does ATP do to the cross bridge once it's bound? Reset, so we bring it, so it detaches, brings it down, and resets it. Because tropomyosin is still shifted over. So it's just gonna keep doing it. It'll just keep going until we take away the calcium. This whole cycle, here we go. Binds, cranks over, resets, goes back. It does it until calcium leaves. So that's the next part. SR calcium. So SR, I'm bad for writing using abbreviations after I tell you guys not to use abbreviations. So, but I just didn't want it to be too wordy on the slide. So the sarcoplasmic reticulum calcium pumps. The pumps use ATP because it needs energy to actually suck the calcium back into it to return it. We have these calcium pumps, bring in calcium. They need ATP to go because you still have way more calcium left in the sarcoplasm. So you're like restuffing it back in. So it takes a little energy. So that's why you need the ATP. It's like a vacuum, bringing it in, stuffing it back into these sarcoplasmic reticulum pillows. See how they're just rotating. So they're bringing the calcium back in. They're detaching. The calcium is plucking themselves off of troponin and they're floating back up into the sarcoplasmic reticulum. And so that requires energy. That's also why people get rigor mortis. If you don't have energy to take the calcium off of troponin, well, your cross bridge is going to stay stuck, right? So again, that's part of rigor mortis. Calcium, troponin unbinds because the calcium's getting popped off, right? And it's going back into the sarcoplasmic reticulum. And then tropomyosin covers back up those active sites. Cross bridges are not going to hang on anymore because they're blocked. And that's what relaxation is. Calcium is back in, cross bridges are unhooked, active sites are covered back up. Now we're relaxed muscle. So that was a sliding filament theory. The thin filaments, remember actin, They're sliding in towards the M line, which happened to be the middle of myosin. The thin filaments are moving over the thick filaments. A better way to describe it is cross bridges are pulling the thin filaments toward the middle of the sarcomere, and the Z lines are coming in together. And that's how that's happening. It's Z lines of each little section that's coming in together. So collectively, it makes for quite a bit of movement. So this is more just word form. So if you, when you get back and you want to kind of map this out and you're like, okay, how's this go? And this, the best way to learn this is, or to review it, is to tell somebody about it. This is the 5 basic steps. So it all comes down to this. Exposure of the sites. Ca means calcium, Tn is troponin, Tm is tropomyosin. So it's going to exposure, involves all 3 of those parts. Binding of cross bridges, that's the myosin lifting up. Pivoting of the myosin heads. Detaching requires ATP to detach it and reset it, and it's going to keep doing it until calcium is pulled away. Relaxation is about plucking off calcium off of the troponin and putting calcium back away into the sarcoplasmic reticulum. So really, calcium is the trigger. We just have all these other parts that are regulating where it's going and what's happening to it. But it's either calcium's out of the SR or calcium's in. And those are the 2 states of whether you're contracting or not. We did a lot last class period. We covered a lot. So I'm going to start off today with, we're just going to kind of recap. So I'm going to put our lecture sort of on this thing, and we'll just go through. I'll sort of select a few questions here. So we've got the muscle coverings. You guys remember those? What's the one around the whole muscle? Epimysium. Okay, there we go. Epimysium around the whole muscle. Then what's the next one around a bundle of muscle cells? Perimysium. Excellent, we have that. And then the last one around an individual muscle cell? Endomysium. And because they're these wrappings, really thin, like with the ends like a Tootsie Roll, all of the ends come together when the muscle has ended to become the what? The tendon. Okay, so that's that component. Then we'll go here. Do you remember the cell components? All right, what is the cell membrane called? Sarcolemma. Lemma? Yes. We right? Yep, there we go. Sarcolemma, outer cell membrane. What is the cytoplasm called? The sarcoplasm. Excellent. So I don't really have an arrow to that because it's just sort of implied that it's the medium within the cells. And then T-tubules. What do they do? What's the job of the T-tubules?
Electrical impulses.
Electrical impulses, but when a nerve is going to come down and release its neurotransmitter onto the sarcolemma, causing the sarcolemma, which the term is depolarized, but we'll put more meaning to depolarize when we do neuro on Monday, cause it to depolarize, which is more activated or electrically electrified, if you want to think of it now. The charge, get it to be positive charge, the T-tubules run that charge, so through the whole muscle cell, even the inner core gets activated. It's not like the outside does it first in the middle, it runs it through so that when finally we go to release the calcium, it's happening from the outer portion as well as the inner portion at the same time, so the muscle will contract together. So the T-tubules sort of make that happen, so it brings that electrical impulse down into the core. Does that sound good? Okay. So the T-tubules will bring this depolarizing impulse down into the muscle to cause, what's the next step that's going to happen if we want it to activate a contraction?
Calcium's going to release.
Calcium's going to release. Where is calcium kept? Here we go. The sarcoplasmic reticulum are these little blue pouches wrapped around all these little myofibrils that's containing calcium. So the sarcoplasmic reticulum is holding in the calcium, and it's really when we're going to contract, the T-tubules send the impulse down to say, sarcoplasmic reticulum, open the gates, open these channels, and calcium will go out. So right now at rest, T-tubules are quiet, sarcoplasmic reticulum just holding on to the calcium. It's nice and close, just hanging out there. So this is just normally what's going on. They're just holding on to the calcium. Then the myofibrils are within throughout the whole muscle cell, but they're the ones just waiting for the signal to either contract. So really when we want to contract, what is the actual contraction process? Who remembers what little arms are moving and stuff? What's going on? That's the myosin, also known as the thick filament. So the myosin has these cross bridges. That's what the little hockey stick is, golf club is. The cross bridges lift up and it's going to bind to what? Actin. So cross bridges are going to bind up to actin, and cross bridges automatically, they lift, bind, and then what? And they pivot over. Yep. And that pivoting over just shifted the actin. So if this yardstick is actin and my arm is, you know, so this is at rest. So if you're like wiggling someone's arm like this, you know, it's like the actin and myosin, just there's nothing in the, they're not connected. So it's just someone loosey-goosey, their arms are all loose. But then all of a sudden, we get to a point, which we know it's because calcium was out there, the myosin lifts up and it binds and it hangs on to actin. And so that's what it wants to do. Hang on to actin as soon as this active site is exposed. So it hangs on to actin, and as soon as it does, it pivots. And now we've moved actin over. So if you imagine lots of these yardsticks connected to a Z line, and another one on this side connected to a Z line, you could see that the actin pulling this way, or sorry, myosin cross bridges pulling this way on actin, myosin cross bridges this way, pulling on actin this way. Collectively, the Z line to Z line, which is a sarcomere, comes together. Do you guys remember, on our actin and myosin, we have actin here and a myosin's here. What's preventing myosin from binding to actin anytime? Tropomyosin is this rope that's wrapped around covering the binding sites, the active sites on actin, the ones that myosin cross bridges want to hook up to and bind to. So when it's covered, myosin's just laying there going, I wish I could have actin to bind to, but I don't see anything to bind to. So it's just there, kind of like hanging out. And so that's when you can move your arms nice and loose, because it's just actin and myosin moving across each other without any connection being made. That's what makes the looseness. So then if you have, so then what controls tropomyosin? So then there's troponin. You can say it troponin or troponin. Either way, I've heard plenty of people say it both ways. So that is the controller of the tropomyosin. So how does it control it? When does it tell tropomyosin, move over? When calcium leaves the blue thing here, sarcoplasmic reticulum, floats out, binds straight to troponin. Troponin moves tropomyosin. Cross bridge is like, there it is, there's the active site, bings up, grabs it, and pivots over, and that's our first cycle. Then how do we relax? Ryan was saying earlier. We need ATP to come in, bind it, it detaches it. Energy, we'll say, this red marker is my energy molecule. So we myosin, we cranked over. Energy helps to bind it and reset, and then we let it go, and then the remnants of that ATP makes its way back to the mitochondria, and we reform new ATP. The ATP is causing it to detach and reset. So you're resetting the hinge. Remember, it's 2 hinge points, but lets it unhook and lay down, and then that head pivots back out. So then it's ready to go. But that's not relaxation. That's just undoing the cycle, because another cross bridge will be bound and it's going to be pulling while this one's coming down. Then when this one's detaching, so the muscle itself is still in a contraction state, even if you have ATP detaching it. And we'll just go hop here to myosin. We'll just do this. So we have the thick filament myosin. So it's showing you in a little bit better way where you have some of the little cross bridge guys spacing you out the screen, some up, some down. If we show you a single one, this is what it looks like. But on a single myosin segment, we have them opposing each other from the midline. You know, so some are pulling on one side, some are pulling it because they're just going to pull together. Yes, because they're going to have the actin here, There. So when these little blue guys come up and hook up, these ones are going to hook up there. They're going to pull in, and so this set of actin goes that way, and that set of actin is going to come into the midline. So you have muscle shortening happening at a sarcomere, but then it happens again at every neighboring sarcomere. So you can see collectively, it's like, you know, an accordion coming in together. The sides are really coming in, but each section is individually being pulled into the midline. So then we talked about it going lifting up and it pivoting. So that's just the natural response of myosin when it sees an active site on actin. So that's why we like to keep it covered up by tropomyosin all the time, unless we only want to contract. So then we need energy to reset it down. In order to do that, ATP is required. So we have to bind it to that. So it's in this up position. It would actually be bound to actin. We just haven't gotten to that part in this part of the lecture. And then it will reset it down, re-splits it into ADP, adenosine diphosphate, and inorganic phosphate, which gets reassembled through the mitochondria, and we can recycle it again. So the actin portion, you guys know here, as we bring it out here, we have the binding sites that we want to keep wrapped up by the regulatory proteins, tropomyosin. There we go. So that's going to be the rope. And then we have the troponin, which is going to be the controller. And in this case, calcium is going to want to go to the controller. Yeah, so what reality is, calcium is going to go to the troponin, which actually happens to be every 7 beads on an actin, but you don't need to know that for your tests, but it happens to be. That's the spacing of it. And so as soon as calcium is bound to troponin, does it do this or something else happen? Tropomyosin there is going to then move, and now it's uncovered, exposing what?
The active sites.
The active sites on actin. So now the active sites on actin are exposed. What would happen? Those little cross bridges want to lift up, hook up, crank over. We need energy to reset it, and it will keep doing it as long as calcium is on troponin and tropomyosin is out of the way. And it will just do that over and over and over again.
So it's like a constant loop.
Yes. And as long as you're making energy and you have calcium keeping that shifted over. That's pretty much how it works. So the motor units, you guys remember motor units. What defines a motor unit?
A neuron, right?
It's
One neuron. Yep.
And it branches off into.
Yeah. So you can have a large motor unit of like 100 muscle cells or a small motor unit of like 3 muscle cells. It doesn't matter. A motor unit is a single neuron going, and however many it may branch, depends on the muscle that you're talking about. So that's a motor unit. So so each muscle cell does have individual nerve activation, but it might be being activated by the same nerve as other cells as well. So that's a motor unit. So if we're talking about the end of a neuron going to a muscle, that is the neuromuscular junction, where the nerve goes in the muscle. And then we'll go quickly here, expand that so we can see the end of the nerve releasing neurotransmitters. And in this case, do you guys remember what the neurotransmitter is for muscles? Acetylcholine, excellent. Acetylcholine to the dark red here is the surface of the muscle cell, so that's called the sarcolemma. So these little acetylcholine guys are going to bind to the sarcolemma, those receptors. We'll go in a little more detail on this when we get to neurophysiology. And then it's going to depolarize, what makes it positive, and this positive is going to kind of fall like dominoes down these T-tubules all the way into the core of the muscle. On its way, when the T-tubules depolarize, it's then telling the sarcoplasmic reticulum, now's the time to let the calcium out. So then calcium then gets released out into where the myofibrils are. Move this along. So it's going to go in there to then ultimately bind to troponin, move over tropomyosin, cross bridges start doing their thing. And when it does its thing, what's going to end contraction? What's going to be the point where we stop? Excellent. So when calcium finally gets plucked off of and removed off of what? Troponin. So it's going to come off of troponin. So here we have out. That represents the top bubble, sarcoplasmic reticulum. Pink is calcium. Calcium is on the troponin. This here. So we have calcium on the troponin, which has made our yellow active sites available so the heads of our myosin have a place to bind to. And this will continue as long as we have ATP and we have the calcium out. We have the sarcoplasmic reticulum calcium pumps. They use ATP, and so these pumps will actually require energy, and they're swirling and they're going to bring the calcium. So it's going to remove the calcium off of the troponin, pluck it off to send it back into the sarcoplasmic reticulum, and then we are going to cover up these active sites again. Because there's way more calcium still in there. So you're kind of having to shove it back in. So it has to go against its concentration gradient, which is why you need the ATP for it. And so it's plucking it. And that is what causes relaxation, is the removing of calcium, unhooking from troponin, tropomyosin shifting back over, blocking cross bridges so it can't bind, and now you're relaxing. And it happens sort of progressively. A little bit happens here, a little bit happens there as the calcium's getting plucked off. So when you are relaxing, it's not a peak force and then zero force. You actually have a decline in force because you're unhooking these different cross bridges.